Friends, you are most requested in Dr. Nirankari video channel, and in this section we will describe about kingdom Monera. In this video link we will describe about sexual reproduction in the organism of kingdom Monera. Sexual reproduction, haploid, diploid stages, gamete formation and fusion are absent in bacteria. In bacteria, in sexual reproduction, haploid, diploid stages, gamete formation, and fusion are absent. However, gene recombination can occur by three methods: conjugation, transformation, and transduction. Our heading is sexual reproduction in bacteria. Sexual reproduction in bacteria. Haploid, diploid stages. Haploid, diploid stages. Gamete formation, and fusion of gametes are absent. However, gene recombination can occur. Gene recombination can occur by three methods. And these methods are conjugation, transformation, and transduction. So we will describe about what is conjugation. Conjugation is coming together of two cells or gametes for purpose of sexual reproduction. Conjugation is coming together of two cells or gametes for the purpose of sexual reproduction. In bacteria, conjugation involves transfer of genetic material. Conjugation involves transfer of genetic material from one bacterium to another, from one bacterium to another through cell to cell contact, through cell to cell contact. Conjugation was discovered by leader Berg. Conjugation was discovered by leader Berg and Tatum in 1946 in case of E. coli. In case of E. coli. Our next heading is conjugation in gram-negative bacteria. Conjugation is gram-negative bacteria. There are two strains of bacteria. There are two strains of bacteria. Donor male and recipient female. Donor bacteria poses a plasmid. Donor bacteria poses a plasmid having fertility factor. Fertility factor or F. Plasmid they are F plus. They are F plus. These bacteria produce 1 to 4 these bacteria produce 1 to 4 long pili called sex pili. Recipient cells are F minus. Recipient cells are F minus without fertility factor. Without fertility factor. When the two types of cells happen to come nearby, when the two types of cells happen to come nearby, the donor cell extrudes a protein Then the donor cells extrudes a protein from the tip of tips of sex pili. It helps the donor cells to get attached. It helps the donor cells to get attached to the recipient cell. To the recipient cell. Wall of the recipient cell dissolve. Wall of the recipient cell dissolves in the region of contact. In the region of contact. 6 pilus forms a conjugation. 6 pilus forms a conjugation bridge between donor and recipient cell. Between donor and recipient cells. Fertility factor or plasmid of the donor. Plasmid of the donor replicate and copy of replicates and copy of a plasmid passage. F plasmid passage into the recipient cell. Into the recipient cell through the conjugation bridge. Cell through the conjugation bridge. Passage of F plus plasmid into passage of F plus plasmid into recipient cell changes it into donor cell. Changes it into donor. The phenomena is called sexduction. The phenomena is called sexduction. Conjugation of this type is called sterile male method. At times, F plus plasmid integrates. At time, F plus plasmid. At times, F plus plasmids integrates with the nuclear body. Integrates with the nuclear body of the cell becomes episome. Episomes and changes the donor cell into donor cell into HFR, high frequency of combination. High frequency of combination. Our next heading is conjugation in gram-positive bacteria. Conjugation in gram-positive bacteria. They do not have sex villi. Do not have sex villi. Donor and recipient a donor cell secrete a protein. A donor cell secretes a protein adhesing over to the surface. The protein helps the donor cell. The protein helps the donor cell in sticking to a recipient cell. In sticking to a recipient cell for conjugation. In Streptococcus faecalis, the recipient cell produce a pheromone-like chemical. Pheromone-like chemicals for attracting the donor cell. Our next heading is transcription. transformation it is the change in genetic constitution of bacterium by picking genes of its dead relatives present in outside medium and integrating the same in its nucleate. It is the change in genetic constitution. It is the change in genetic constitution of a bacterium from picking gene of its dead relative, picking genes of its dead relatives present in outside medium, and integrating the same in its nucleate. Transformation was discovered by Griffith in 1928. The next topic is the method of sexual reproduction is by transduction. Transduction. It is the process of carrying over. It is the process of carrying over genes from one bacterium to another through the agency of a virus. Transduction was discovered by Gender and Lederberg. In 1952. In case of Salmonella typhimurium. In case of Salmonella typhimurium. In transduction, a virus picks up one or more bacterial gene instead of its own gene. Genes, as it attacks a new host. The genes are passed into the new host. New host. The transducing virus is seldom harmful. The transducing virus is seldom harmful because it does not carry its full gene complement. Transduction is of two types. First one is generalized and restricted. Any gene is transferred in generalized transduction while the same particular gene is passed in. And in case of restricted or specialized transduction, it is successful if the new gene integrates with the host gene, or abortive if the genes are fails to do so. In our next video link, I will describe about mycoplasma. So wait for my next video link, and next video link I will describe about the mycoplasma and three kingdom division of organism, archaebacteria, then groups of archaebacteria, methanogens, halophiles, and thermoacidophiles, and then I will describe about economic importance of bacteria. So please wait for my next video link, and if my material is according to your aim, then you are requested to like, subscribe, and share my links to your in your friends groups. Thanks.